So this is a picture of a neuron, which is a nerve cell, and they are most specialized in animal cells. There are three types of neurons. There are sensory neurons, interneurons, and motor neurons. The sensory neurons transfer information from the receptor to the brain or spinal cord. From there, the interneurons integrate the information inside the brain or spinal cord and send the information into the motor neuron that transmits information to the muscle or gland. So now let's look at how the signal travels through a neuron. A signal has many entry points through the dendrites, which are branched extensions that receive signals from other neurons. A signal then travels into the cell body, which contains neurons organelles. Then the signal transfers through the axon hillock into the axon, which is a longer extension of the neuron. The axon coated with Schwann cells makes the myelin sheets, which by insulating the axon speed up the signals. This leads the signal to jump from node to node, which is the process called salutary conduction. The transmission of signals through the axon are like dominoes, where the signal starts in one area, opening the protein channels, and moves down the axon. Then the signal reaches synaptic terminal, where the signal is passed from the presynaptic cell to the postsynaptic cell through neurotransmitters, which will be mentioned in more detail later in this video.
Action potential begins with the resting potential, the membrane's potential characteristic of a non-conducting excitable cell, with the inside of the cell more negative than the outside. Even though both the sodium and the potassium ions outside and inside the cell are positive, neurons actually have a pretty strong negative charge inside them. This is due to the presence of other molecules, called anions. They are negatively charged, but are too big to leave or diffuse through any membrane channel. At step 2, the stimulus reaches the threshold potential, or the potential that an excitable cell membrane must reach for an action potential to be initiated. The action potential is an explosion of electrical activity that is created by a depolarizing current. This means that some stimulus causes the resting potential to move toward 0 millivolts. When the depolarization reaches about negative 55 millivolts, a neuron will fire an action potential. During depolarization, which is a change in the electrical state by which the inside of an excitable cell is made less negative compared to the outside than at the resting membrane potential, the sodium ion channels open and the potassium ion channels close. A neuron membrane is depolarized if a stimulus decreases its voltage from the resting potential of negative 70 millivolts in the direction of 0 voltage. At the hypershoot, or the peak action potential, the voltage-gated sodium ion channels begin to inactivate and the voltage-gated potassium ion channels begin to open. This initiates repolarization. Repolarization at step 5 is the change in the membrane potential that returns it to a negative value after depolarization changed the membrane potential to a positive value. With less sodium ions moving to the cell and more potassium ions moving out of the cell, the membrane potential becomes more negative, moving towards resting value. The undershoot, or the hyperpolarization phase, is when the membrane becomes more negative than when it was in the resting state. Here, the potassium ion channels close slowly as the movement of potassium ions out of the cell ceases.
This is a picture of the voltages of a nerve cell. The voltage gradient is established when sodium diffuses outside of the cell where the charge is positive outside and negative inside the cell. Sodium and potassium channels open and close after an all-or-nothing response in which the nerve cell reaches threshold potential. Each channel is used to establish a voltage and concentration gradient in the nerve cell by allowing sodium and potassium to diffuse through the cell, only at certain voltages through passive transport shown in the picture. Through sodium-potassium pumps, the nerve cell uses an active transport protein in the membrane to pump out 3 sodium ions and pump in 2 potassium ions to establish a concentration gradient, with sodium on the outside and potassium on the inside, demonstrated by our picture of the salty banana. This process requires a large amount of ATP to complete this process, which is around 25% of the energy we consume every day.
Synapses are the location at which the action potential is transmitted between neurons. Electrical synapses contain gap junctions that allow electric currents to flow directly from one neuron to another. These synapses are used to coordinate the processes carried out by neurons in a large variety of organisms' daily lives. The majority of synapses, however, are chemical synapses, like the one in this image. Neurotransmitters are synthesized by presynaptic neurons and packaged in membrane-bounded compartments called synaptic vesicles. These neurotransmitters can be excitatory and bring the membrane potential toward threshold, like dopamine and glutamate, or inhibitory and move the membrane potential farther from the threshold, like GABA or glycine. An action potential at the synaptic terminal results in the opening of voltage-gated channels, which in turn causes the neurotransmitters to be released. The neurotransmitters diffuse across the synaptic cleft, the narrow gap that separates the presynaptic neuron from the postsynaptic cell. The postsynaptic membrane reacts accordingly to the neurotransmitter based off of either its general responsiveness or the concentration of the neurotransmitter.